Good afternoon, everybody. Good afternoon, Kostya. Kostya, my presentation is not about poliovirus. I never worked with you, as you know, but I always worked near you and always shared with you most exciting things. And most important thing is that you were one of those people who paved my way into virology, because I don't know you as long as Vadim ago, but we know each other almost 50 years, which is still a lot of time. And for those of you who don't know, when Eugene and I became undergraduate students in Moscow University and became members of the virology department, Kostya, who was at that time an early stage graduate student, was the supervisor of our group. So he was a really good supervisor. We didn't see him too much. And he did, but he was inspirational enough because one of the lessons I learned from Kostya is that there are no limits. You can do whatever you want, and you always should never hesitate. So, first thing about Kostya, just a few words, is that he does not age. I think you all appreciate that, and it's obvious. Another thing, he's charming and versatile. And another thing is he's equally strong in mechanistic and discovery studies. And of course, he's a big virological magnet, as you can see from this parade of great people who are near him and a parade of people who are sitting in this room. So, my story, Kostya, is to you, not to this audience, because I was thinking, what can I say which may entertain you? And since you have been also looking at viruses as enemies and you try to protect yourself and us from that, I decided to go with you and look at them from different angle, which is something we, as virologists, we usually never think about, although some of the things I'm going to say are obvious, but we're not thinking about them, and some of them are very new and have not been yet published. So, I will be talking about half of our DNA, which is occupied by highly repetitive elements, vast majority of which are actually viruses or products of viruses. These viruses, vast majority of them originate from retroelements and amplified through the mechanism of reverse transcription, and invaded our DNA in our predecessors millions of years ago as a series of multiple explosions. So we all know what reverse transcription is, and these elements belong to two major categories, signs and lines, short and long interspersed repeats, and they are amplified through a mechanism which resembles PCR, going through RNA and then back into DNA. So, why I want to mention them? For a very interesting reason, which again, most of us don't think about. If you look at the time when they originated in our genome, and right now we're speaking about short elements like ALU sequences or B1, B2 of rodents. If you look in the phylogenetic tree, you realize that sign elements invaded into the DNA of common predecessor of major orders of mammals. So, for example, all primates, regardless who they are, lemurs, chimpanzee or human, or green monkey, have ALU sequences. And all rodents, regardless who they are, they have B1, and some of them have B2. So, this means very interesting kind of evolutionary perspective, or I would say retrospective. So if you look at the paleontological evidence at the time when dinosaurs still occupy this planet, and you will try to make a zoo of mammals, that zoo would be pretty boring. Because at that time, mammals were pretty much the same. There was no this diversity as we have now. So then there was an interesting period of time when the massive extinction of dinosaurs, and massive appearance of huge number of different archetypes of mammals. And suddenly, within a relatively short evolutionary time, the zoo would become much more interesting, and which we enjoy today. So all this great variety of morphologies appeared as a result of some kind of evolutionary period of tremendous creativity, after which there was a long period when there were no major evolutionary inventions in terms of morphology. So what is interesting is that if you look what happened during this time, you will understand that it is exactly the time when sign elements appeared and exploded first time in the genome of our ancestors. And since this signs, every order of mammals has its own sign element, you immediately start thinking, what if these explosions were creators of this morphological diversity? In fact, if you have 1 million copies of ALU and all of them are in intergenic spaces or in introns, it means that those of them who acquired them in open reading frames are dead. So which means that this explosion potentially could be lead to genetic catastrophe. And those species who had compact genomes during this period had very little chance to survive. And maybe the fact that we carry such a huge genome with such lots of unnecessary kind of space may be simply a result of the fact that we are a progeny of those who survived because they were not killed by this process. Another thing is that what if, there were plenty of other things associated with this process. For example, we have to prohibit homologous recombination against repeats because otherwise karyotype would be killed immediately. But one very important question was, does this archetype diversity could be led by viruses? I can tell you that answer is yes. It is a hypothesis, but this hypothesis is proven now by the experiment which humans did on dogs during last several hundreds of years of dogs' evolution under selection of selectionists. Because we all know that the dog represents the mammalian species with the biggest interspecies range of diversity. There is nothing else. All other domesticated animals never showed this diversity. So the question is, what is so different among dogs, genome versus others? And today, if you look in literature, and you will find all these events have been cloned and sequenced and mapped. What is the genetic event which determines short leg of corgis or dachshund? What is the genetic event which causes the flat nose of a boxer? Every time it's integration of sign or line element in the new place of genome. Which means that viruses plays the role of actually morphogenes creating lots of freaks from which either natural or artificial selection picks up new creatures with new appearance. So which means that the fact that there is such a diversity of mammals is not the only result of adaptation to certain niches, but because of creation of huge number of freaks during explosions of sign elements, and those of them who could find the niche to survive give rise to current inhabitants. So now the next question I want to say, so I think it's a remarkable conclusion which people usually don't think about. Another question is, can we look back and trying to retrospectively restore the activity of I named this plethora of retroelements in the genome word retrobiome. I think it deserves the term. Half of the genome deserves the term. So, and yes, it's possible. We did a recently work which we have not published yet, but it brought us to very interesting observations. It's very hard to look at the intensity of amplification of different parts of retrobiome looking at the repeats themselves. They are too abundant. But as a side product of amplification of line elements and sign elements is the appearance of pseudogenes, process pseudogenes, because sometimes the reverse transcriptase makes copies of mRNA, and you can find these copies in the genome. We have thousands of them in our DNA. This is intronless, promoterless copies of genes. And they're much less abundant, and you can measure their evolutionary age simply by looking how close they are to their own mRNA. The longer they exist in the genome, the further they evolve due to neutral acquisition of mutations from their ancestor. And what we did, we simply took the plethora of the array of all pseudogenes we could find and classified them according to the degree of their diversity from their own mRNA, which is basically a time machine, which shows, and then you look what is the proportion of them in the genome. And of course, if you take, let's say, 3 species of rodents, mouse, rat, which are obviously quite close, and a naked mole rat, the notorious animal with an endless lifespan, more than 30 years. You look at them and you do this histogram, and you see that indeed those pseudogenes were actually unique for rat and mouse. They are all over the place in terms of degree of diversity for mRNA, but those which are common and which appeared at least minus 10 million years ago, they all pretty far diverged from their own mRNA, indicating that our time machine works. And then we looked at two things. First, we look, laboratory mouse has lots of pseudogenes which are very identical, completely identical to their own mRNA. That means that maybe the process of new pseudogenic appearance goes as we live. So what we did a strange thing. We took 12 individual mice from C57 black and sequenced them. And we found pseudogenes which were different even with this very small number of animals, and indicating that indeed in this Charles River population of C57 black mice, which are maintained in a very high degree of inbredness, there is ongoing process, and we caught it in the act. But there is another thing. Another most striking thing we found when we looked at the naked mole rat, that fantastic rodent which does not have limit of life, so which lives at least 10 times longer than the mouse. You see that it does not have at all any pseudogenes which would be identical to their own mRNA, indicating that the amplification, the activity of that retrobiome stopped several million years ago. Whether it has any connection with the exceptional longevity, I don't know, but it obviously suggestive that some time ago, this particular, you see how many copies they had in several million years ago. So a common predecessor of this, ancestor of this and other rodents, at some point passed through the period of evolution when there was a very strong activity of retro elements, which probably led to significant extinction. But those who happened to survive somehow stopped this process and today enjoy A, existence, and B, almost eternal life. Whether these things connected or not remains to be shown. So this is obviously, and if you compare the number and the proportion of pseudogenes which are identical to the mRNA among different species, you see that naked mole rat is clearly an exception. So that leads to a very provocative idea that maybe together when we accepted sign elements in our DNA, we together accepted the clock of life, and maybe this remaining activity of these elements somehow play the role of a clock, and maybe retro elements are driving our aging. And if it is true, then aging is a viral disease which technically can be treated by antiviral treatments. I'm not saying that I'm sure about that, but it's an interesting, inspiring direction of research. So, what else viruses can do? Now, very interesting question, viruses and cancer. We all used to think that viruses is a risk factor, that they're drivers of cancer. We are all what Zilbert did in that regard, and we know that viruses also carry oncogenes and so on and so forth. We know that currently, I don't know, EBV is associated with cancer and so on and so forth. But in reality, if you look at viruses in a more broad way, also taking into account those which are living in us and occupy half of our genes, you will find that situation is not that simple. If you have somebody in your genes for millions of years, you not only allow them to be there, but they sometime to exist probably give you some advantage to take care of their own existence. So what can we say in that regard? Well, the story started, of my interest in that started from the paper we published 10 years ago, in which we demonstrated that p53, the tumor suppressor, it actually plays a very important role in negative epigenetic control of expression of retro elements. So this finding was confirmed by many since then, and we came up to this following conclusion. So these repeats, being a technically dangerous content in our DNA, is locked there epigenetically at several levels, one of which is p53-mediated epigenetic silencing at chromatin level and DNA methylation. So if, however, this thing does not work, what happens is that the cell recognizes activation of these elements as an invasion of viruses and activates interferon response, which plays dual role. It either kills the cell directly through necroptosis or attracts immunity to eliminate the cell. So in this regard, retroviruses and retrobiome plays the role of a tumor suppressor because it directs immunity towards the cells which are in danger of losing epigenetic control. So this is their kind of Dr. Jekyll's appearance. Well, however, tumors we know appear, and all tumors, with very few exceptions, live under constantly active transcription of retrobiome, which means that they somehow learned how to live without that, most likely simply reducing sensitivity to interferon and acquiring as a result sensitivity to oncolytic viruses. This model explains why oncolytic virus phenomenon exists in principle. But when cancer carries this, it acquires genomic instability, inflammation, and sensitivity to oncolytic viruses. Why? Because this element, line 1, which occupies about 20% of our DNA, and 150 copies of which are still intact in it, out of 500,000, when it gets activated, it basically leads not only to new insertions, which probably is a very minor role in its mutagenesis, but it also encodes endonuclease, which makes holes in DNA and through that stimulate point mutations. Every new copy of integration becomes the hotspot for homologous recombination, and I strongly believe that such thing as chromatripsis or complete disruption of karyotype in some cancers may be driven by these insertions and the recombination. It leads to constant inflammation, and all this, of course, significantly impacts cancer phenotype. So, how does p53 activity looks today through this knowledge? I think it's a tremendous revision. This is a textbook knowledge. Our cell populations are under constant pressure of mutagenesis, either through bad habits, like on the top, or through endogenous activity of retroelements, okay? But then, when in population, the cell which happens to have broken DNA, being punished by p53, which acts as a positive transcriptional regulator, which activates genes of either apoptosis or senescence, taking away these cells from proliferation. This, so you see, plays the role of a cleaner, of a punisher. But this is textbook knowledge. This new knowledge tells that p53 has another role. It's a repressor of intrinsic mechanism of mutation generators, which means that it plays the role of a Dutch boy, which with its finger saves the country from a flood. And when there is no p53, there is not only this punisher who does not allow this cleaning job to do, but it also, there is no more Dutch boy, and you have a flood of events happening as a result of activation of retroelements, which obviously leads to lack of control over accumulation of variability, and leading to notorious cancer ability to evolve and adapt to pretty much everything. Why this picture is interesting? This picture is interesting because it shows why our ability to extend our life without cancer through healthy habits really fails, even though we all try to do that, but without not very big success. But most importantly, it gives you a very interesting perspective that if you could stop retrotransposon activity, you potentially can reach stabilization of the genome. So basically what you can expect that maybe you can, by pharmacological inhibition of retrobiome, you can partially restore genome stabilizing function of p53. So, and we tried to do that in the following experiment. This paper has been just published. So we took the mice with breast cancer, spontaneous model, and we put them on the drinking water with inhibitor of reverse transcriptase, which inhibits line 1. So, and stabudin. Well, cancer occurrence was exactly the same. It did not affect appearance of cancer even slightly. But if you do chemotherapy of these tumors and they all respond, but they all then relapse, all they come back, become resistant. But if you take these tumors and together with chemotherapy, you give them in drinking water inhibitor of reverse transcriptase, you now have 3 times increase in progression-free survival. meaning that you, for the first time in oncology, you have the drug which has zero anti-cancer effect directly, but can block cancer creativity or cancer ability to adapt and reoccur. This idea is now currently under clinical testing. We are running clinical trial in small cell cancer patients who are on top of standard of care receiving inhibitors of reverse transcriptase. So, which means that you can look in this paper if you are interested. The mechanism appeared to be very interesting because initially we thought it's going to be very primitive, just these elements create genetic diversity and from which selection is picking resistant variants. Results appeared to be different. Cells become resistant to line 1 activation because they activate pro-survival interferon and NF-kappa B pathways and survive treatment. And a reverse transcriptase inhibitor suppresses this not because it blocks retrotranspositions, but because it blocks inflammation. So, and which means that the same mechanism which drives cancer preventive function of retroelements, when cancer is already established, plays the role of an adaptation mechanism, which is druggable. Well, to summarize, in cancer, retroelements, viruses which live in our DNA, they work as cancer preventive agents by enforcing immune surveillance to the cells which happen to block control of their epigenetic stability. And however, at the same time, they drive treatment resistance to already established cancer. What is good, that both of these things are druggable. Finally, in the last minute, I will tell you something which I'm for the first time stepping in the territory of Kostya. And Kostya's territory, of course, is vaccinology and the ability to use vaccines to prevent us from disasters associated with viruses. So what can we do in that regard? So let's think about that. So this is the patient with a tumor. The tumor activates line 1. Line 1 encodes 2 proteins, including reverse transcriptase, but these elements are not active in any normal cells, which means that by activation of line 1, the organism is supposed to consider this as the appearance of cancer-specific antigens. So the questions we want to address, do we see signs of anti-tumor immune response in patients with cancer? And what we did, we took about 4,000 serum samples from 4,000 cancer patients, different type of cancer, and we measured titers of antibodies against line 1 antigens. This paper has not been yet published, it's in review right now. What we learned from that was mind-blowing. So I will just show you some highlights. So this is, those of you who are experimental biologists will appreciate why I'm showing this data. These are HeLa cells with inducible line 1. If you do add doxycycline, the cells activate line 1 antigens, and you can stain it either by immunofluorescence with rabbit anti-ORF1 antibody, or on Western you see the single band of ORF1. So, but now what you see here, these are four pictures, they stand for immunofluorescence staining of similar cells with just the blood of a patient. See, these are ovarian cancer patients. No antibodies were isolated there. It's just 50,000 times diluted serum of cancer patients. You see how beautifully specific it works only in the cells following line 1 activation, and see how cleanly it gives you just a single band on ORF1 in the lysate of cells when you activate it by doxycycline. So, which means that anti-ORF1 line 1 antigen response is by far the strongest autoimmune response which we ever seen. The levels of these antibodies is amazing. So in some patients we have to dilute it millions or billions of times to reach dynamic range in the ELISA. So, and when we looked in which cancers do that, it appears that these 5 cancers, ovarian, lung, esophageal, liver, and pancreas, the cancers with the least capable we detect early are those which have the highest levels of these antibodies. So, but this study, which also brought us to a very interesting observation, it appears that we all have antibodies to line 1, just low titers. It's not plus and minus. It's not like cancer patients have it. So it very nicely fits the idea which I gave you in the beginning, that maybe anti-line immune response is constantly protecting us from cancer. And these levels of antibodies which we have simply may be recollection of successful anti-cancer effect of anti-line 1 immunity. And only when the tumor learns how to live without being killed by immune system, this titer starts going up. So all this together brings me to the final thing. So viruses and their products occupy about half of our DNA. Virus-driven mutagenesis has defined our appearance, how we look. Virus is likely to contribute to mammalian aging through DNA-damaging, epigenetic destabilizing events and the inflammation-inducing. So epigenetic desilencing protects us from early cancers because it attracts immunity to these cells. And viruses of this kind are great targets because they are driving treatment-resistant progression, but they can be suppressed by reverse transcriptase inhibitors. Kostya, thank you for inspiration.